So I'm going to redo a couple of slides. We may need to backtrack from where we were yesterday. I just want to be talking about this and making sure I was explaining it appropriately. I saw some head spinning yesterday, and so I don't know, which is maybe that's a spinal reflex. I don't know. It's one of those things. This is the cortex gets overloaded, and then all of a sudden it causes. I don't know. Anyway, so let's look at this. Let's look at what happens in the spinal cord itself from a muscular standpoint. So again, we have that cord gray matter, right? And so this is where all the integration actually comes from, right? So whether you have the brain telling the arm to flex, that's going to be coming down through this cord gray matter. Whether you have these reflexes that are coming about from, say, some of these sensory fibers that are coming in from the muscle itself, right? Because remember, we were talking about how the muscles can sense basically the length of the muscle with those sensory fibers, right? We'll cover those again as well. Basically, there's going to be all these interneurons here, these interconnections that allow for these integration of signals to ultimately determine, okay, what do we want to do with this muscle? We're actually going to be able to, we're going to contract it, we're going to relax it, what's going to happen here, right? And again, this is going out to all sorts of different muscles, and these are activating specific motor units in some cases, so you may find that some of them may be for very fine control, say like the hands versus maybe moving the whole quadriceps. It just depends on what the signal is going to be here, right? And again, all this is going to be heading out through the anterior motor neurons. So he's going to be traveling out here, right? So again, you have all this integration, and it's going to be traveling out here. And we'll talk about more of the higher functions here, some of the higher signaling that happens in just a little bit later. But I just want to kind of focus on the cord first, and then we'll kind of go up from there. So, and remember, and kind of at a baseline, the muscles, unless you're telling a muscle to do something consciously, it typically wants to maintain homeostasis just like anything else in the body, right? So again, if I were to say stretch out the muscle, what does the muscle want to do to counteract that? Wants to contract it, right? If it were to get too short, it wants to do what? It would try to extend it back out by loosening it, right? By trying to decrease the contractions that actually happen there. So this is kind of what we're getting at with talking about these muscle or these sensory fibers. They're going to be kind of located in these intrafusal fibers, right? So imagine these are going to be located all throughout the belly of the muscle. These are going to be able to tell us basically what is happening as far as the size of the muscle goes, right? And again, just like going back to our example of looking at the reflex that we can trigger by, say, hitting the patellar tendon, what does that cause? So it's going to cause the quadriceps to lengthen, not a lot, but it'll cause it enough to lengthen, and then it does what as a response to that? It's going to want to contract again. That's why you end up seeing your lower leg jerk, right? So that's what's basically happening there, is that by extending that muscle, by lengthening it, it says, hey, wait a second, no one told me to do that. I need to go ahead and make sure I get back to normal, and it's going to cause a reflex contraction, okay? Same thing would happen if you had, say, the muscle get too short all of a sudden. You'd have a reflex sort of relaxation to allow for it to get back to that normal size. Because again, it's okay if your patient's kind of sitting on the table and you can elicit the reflexes, but if I'm standing up here, I probably don't want my quadriceps just to go out all of a sudden, right? I probably want to stay standing for the most part, unless I hit my head really hard, then I don't have to give the lecture, I guess, but yeah.
So why do some healthy individuals not have that reflex, or less than others?
So that can all go with the signaling and how it's going to be getting integrated up with the rest of the CNS, right? So we're going to see there's lots of signal pathways that happen here. This is kind of the very baseline, the kind of the most direct sort of reflex you have here, but it gets more complicated as we go talking about things like the cerebellum and the cortex and all of that, which is what we're going to get to in a little bit later. Sometimes some people are just going to have stronger reflexes than others. Sometimes it can be related to things like electrolyte shifts. It can be related to disease. It can be related to all sorts of things, right? So unfortunately, not one clear answer, but again, this is just the basics. We're going to expand out from here. Okay. So anyway, so remember, we're going to have the alpha motor neurons. That's what's going to actually cause the main muscle to actually contract when it's being stimulated, or to relax when it's not being stimulated, as the case may be. And remember, these are also going to be integrated with these gamma motor neurons. So remember, right here in the middle, when you have these sensory fibers, there's no actin myosin actually here, right? So again, this middle portion where the sensory fibers are lying actually cannot cause any sort of contraction itself, right? Even if you were to stimulate it, it would not cause any contraction, simply no actin and myosin. Again, here's another picture showing the same thing. Here's the motor neuron. Here's where you actually had the spindle fiber here. The green is going to be actually what those, basically what these sensory fibers are looking like in yellow right here, okay? So just a different way of looking at it. So the big thing to note here is that when you have, say, the brain says, let's go ahead and contract that muscle. So for instance, if my brain is telling my bicep to flex and to contract, do I want to have that reflex to shut that down, to say, well, wait a second, the muscle's getting shorter, let's go ahead and lengthen it back out? No, because my brain is taking the overriding control there and saying, hey, let's go ahead and contract that muscle and shorten it, right? So again, there's a way to shut down that reflex, and the main way that happens is because with my brain telling those alpha motor neurons to contract the muscle, what are those gamma motor neurons doing as well? Because normally what you would expect to happen is if you have the alpha motor neurons that are contracting the muscle should get shorter or longer? Should get shorter, right? And then that would do what to the sensory fibers here? Shorter or longer? So again, taking the gamma motor neurons out of the case here, right? So again, yes, it will get shorter as well, right? Because again, as the muscle gets shorter, that gets shorter too, right? And that will elicit a response saying, hey, wait a second, the muscle's too long or is too short here. Let's go ahead and send a signal back out to relax the muscle, okay? That's part of that reflex. Well, if we're telling the brain, or the brain's telling the muscle to go ahead and contract here, we don't want that to happen. So what happens is you'll have the gamma motor neurons here, which are going to locate on either side of the actual spindle fiber, is going to, as the muscle contracts, this is also going to contract as well. Right? So this will cancel out the shortening of the muscle there by causing this to actually lengthen out, right? So it's basically kind of pulling the muscle spindle on either side to lengthen it back out to basically keep it at a normal length, right? To keep it at, basically there's no change there, so no feedback response, no reflex is actually elicited there. Make sense? Okay, similarly, if I were to send a signal to the alpha motor neurons that says to relax the muscle, right? Should probably lengthen the muscle back out. Normally you'd expect the spindle fibers and the sensory fibers to be stretched out as well, but you don't want that to occur, so the gamma fibers are also going to relax, and that will then shorten it back up to keep it at a relatively normal length, right? So that way there's no real change to the spindle fibers when I'm having a conscious control saying, hey, I want this muscle to contract, right? However, if I were to say have no control of the muscle whatsoever, and then I were to have, say, a reflex or something come along and stretch that muscle, that's when you see the reflex actually kick in and prevent that from occurring, okay? Or prevent those rapid shifts in size or in length from occurring. Okay, everyone with me so far? Starting to see head spin again, but I'm not going to cut class this early. We're going to keep going. But basically, this is what helps maintain that tone of the muscle, right? So again, we say the tone of the muscle is basically what's that baseline state of contraction, right? So again, all muscles, you shouldn't have any muscles that are completely and totally flaccid. They should have some amount of control. So again, every muscle is getting hit with some amount of action potentials, keeping it at that certain tone. But again, when you have those rapid shifts, that's where we want to prevent those rapid changes from happening. This is what this reflex arc is preventing, okay? Again, going further, looking at the cord, there's all kinds of different interneurons that are going to be here. They're located within the gray matter. Again, they're very highly excitable. It's very rare that you have fibers that are actually or signals being sent down from the brain and actually innervating those motor neurons. Those interneurons are usually going to be the intermediary here. They're usually going to be the middlemen. They're going to be getting signals from both the sensory inputs, from the inputs from the brain, and inputs from everywhere, and trying to figure out, okay, well, what do I actually want to have? What's the total sum here? Say, do I want to contract this muscle or relax it? Whatever the case may be, right? Again, I mentioned some of the other cells. A lot of these are useful for helping coordinate, moving from different areas of the body. Some of them are helpful for making sure we get a very precise sort of signal being sent. That way, we only have those certain muscle fibers being activated when we want them to be. So again, those are just kind of accessory cells. You'll see. Okay. So again, we mentioned muscle spindles and how they're going to be operating here. Remember, we also have Golgi tendon organs, which we're going to talk about in more detail in a second. This is just going to be located on the tendon itself, and this is going to be able to detect the stretch there as well. Because again, the muscles are pretty strong, and in fact, if you had a muscle contract with, say, your quadricep, if it contracted with all the force it could, what do you think would happen to that tendon? It'd rip it right off, right? You have an avulsion there, and we typically don't want that to happen, right? And I've never seen like someone like rip a pec or have like a quad or something like that rip. It's not pretty. Yeah, it's not good, right? So again, those muscles just go and they just kind of sink back, just kind of suck in because they don't have that tendon there attaching it to the bone. Google some videos maybe when you're not like in a just fed state. It's kind of queasy, nausea inducing, but anyway, it's not good, right? So again, this is what we're going to see the Golgi tendon organs are going to help us with. And again, this is good for intrinsic control. These are going to operate subconsciously. A lot of these reflexes are very short. You're going to see it's going to travel right up to the spinal cord. That sensory input is going to be being able to put in on those motor neurons pretty much directly, right? Okay, so again, the central region here of the spindle fibers are not going to have any sort of actin-myosin, so they're just non-contractile. Again, the gamma motor neurons are going to be connected on either side. These again are going to try to prevent sudden changes when you're actually having the alpha motor neurons causing contraction or relaxation of the muscle there. So that's kind of the baseline point there, okay? Again, as you have the spindle receptors get excited, it's usually going to be when you have them being stretched out. If they get more short than they normally would be, that's when you're going to see the signals will go down. But again, changes in action potential, the actual change in frequency is enough of a signal for the spinal cord to take that as a hint, right? So again, if all of a sudden it gets, it'll be sending a baseline level of action potential. So if it gets to be a lot more than that, that probably shows the spindle fibers are being excited, being stretched out. And if it's getting a lower number of action potentials than normal, then it would show, okay, well, probably they're getting shorter because they're not being as activated as much. Okay. Remember, there's a couple different fibers that are here, so there's kind of differences when you're having length changes in the muscle itself. So again, you can have very rapid changes, or you can have kind of slower changes. Slower changes, we're going to be called these kind of static sort of responses. very quick changes in muscle size are going to be what we call these dynamic responses. And basically you have different types of sensory fibers that can kind of detect these sort of things. This nuclear back fiber is better at detecting really sudden changes, really rapid changes in muscle size. It is able to send those signals back to the spinal cord to say, okay, let's have a reflex here. Let's go ahead and make sure we try to prevent this sudden change in muscle size, right? When you have just slow changes in muscle size that are not very quick, you're going to find that both of these fibers will be signaling off at the same time, roughly. And that's going to send, again, a signal to the spinal cord saying, okay, we're having slow changes. Maybe we don't need to trigger off such a strong reflex. Maybe it can be a little bit of a slower reflex or a less, lesser in strength, so to speak. So again, these are just different ways that we're able to kind of damp that response depending on how big of a change we're having in the muscle length. Yes, sir.
So what's the difference between the dynamic response and the gamma fibers that you have in the center?
Just remember the gamma fibers are really only going to be activated when the alpha motor neurons are activated as well, right? So again, that's just really trying to show that when we're having conscious control and I'm saying, hey, I want to contract my bicep, by causing that bicep to shorten, I don't want to have a reflex that says, well, to loosen it, right? Because if I'm trying to lift up something off the ground or something, I don't want that reflex to kick in, okay? And so again, by having the gamma motor neurons firing at the same time as the alpha ones are, you're causing the length of the spindle fiber to not really change at all, so no reflex is really being triggered.
So that's more so like a voluntary response? Like
Primarily, yeah, primarily you're going to find it's more of a voluntary response. Okay. All right. So again, looking at a stretch reflex, imagine if I were to say were to hit the patellar tendon, you're going to see that initially. It's kind of interesting looking at this graph here. Basically, this is comparing, say, like a normal sort of reflex versus, say, an animal that had part of the spinal cord resected. And again, I was reading through the book, and like all these mice having to have different parts of their brain taken out or spinal cord cut, just to like illustrate these differences. I was like, I feel really bad for those mice. Probably have like a mouse uprising if they weren't so like debilitated from all the surgeries we're providing them. But anyway. So again, you're going to find that when you have a sudden change, you're going to find that we are sending back those signals very quickly through the spinal cord back into the muscle itself. So again, when I lengthen out the quadriceps by hitting the patellar tendon, you should have that reflex back that causes it to contract, because that will help to keep that muscle spindle, the spindle fiber back at the normal length, right? It's going to lengthen out and then it'll kind of go back in as that muscle contracts. You notice that there's kind of a dampening effect that happens here, so that way you don't get really kind of jerky movements of the leg. You kind of one smooth jerk, and that's kind of it, essentially. If you actually have an animal that had part of the spinal cord resected, you'd actually find this very, very jerky sort of movements that happen here. So again, it's good that we have our brains intact and our spinal cords intact to make sure these reflexes are nice and smooth. Anyway, again, typically when you have a sudden change, imagine you hit the patellar tendon like that, that will lead to initially a dynamic response, because again, that's when you're having that really rapid shift and change. And then as the movement starts to slow down a little bit, as you start to see that the actual stretch of the muscle start to die down, that will lead to more of a static response. So that way, initially those nuclear back fibers are the ones that are being stimulated most, and then it'll kind of die out and they'll both be kind of being activated at the same time, roughly. Okay. Okay, so as I mentioned, with voluntary activity, typically you're going to have the alpha and the gamma being activated at the same time, so that way that will prevent changes in this spindle fiber length, so that way you do not have any unnecessary reflexes than you want to. Okay. And then you're also going to find this is also good for helping to stabilize joints and things like that. This is why it's good for like postural muscles or like anti-gravity muscles, try to keep people upright. It's good when you have any kind of like sudden shifts or anything like that, you're able to keep yourself nice and upright. Some of the things you can see. So again, looking at different sensitivities to stretch reflexes, you can find some of them are going to be more likely to die out quicker than others. So for instance, if I were to have like the patellar tendon being struck here, you can see this knee jerk that eventually will kind of die out. Once you kind of have that signal being sent up, especially to the brain, that can start to communicate, be like, okay, well, I realize this is kind of not a problem. I just realized it's kind of a temporary issue. Then you'll find that those reflexes will die out pretty quickly for the most part. However, you can have cases where you have this kind of oscillation or jerks that happen here, and this is called clonus, and you can see this with the ankles. And so the way the book kind of describes it, if you have someone who's kind of standing on their tippy toes, right, and all of a sudden they go back down on their heels, what do you kind of notice? You notice you just kind of like, you stop, or do you kind of bounce a little bit? If you do it yourself, you notice you kind of have a little bit of a bounce there, and that's basically because as I am relaxing those muscles there, you're going to notice that the calf muscle is going to be getting kind of stretched out, and that's going to cause a reflex, right? So as that calf muscle being stretched out, what is the reflex going to be? To contract it, right? So it's going to contract the muscle back up, so you have it come back up a little bit, and then you have a smaller reflex. As you go back down your heels again, they'll stretch it out. It's a little bit less strong, but then you'll notice it's still going to have that reflex there. So when you see that, you can actually see ankylosing that can happen in patients. This is actually one of the things we look for. Certain drugs can actually affect this to make it more pronounced than other patients, which I won't get into here. But just know that when you see clonus, that's what they're basically talking about, is this oscillation of muscle jerks that happen when you're having kind of sudden changes in length of the muscle. Okay. Okay, so that's it for the spindle fiber. Here we're going to talk about the Golgi tendon reflex. Now, typically here, this is really just trying to prevent us from ripping out the tendons from the bones, essentially, right? So again, this is only going to have an inhibitory effect. Whereas with the spindle fibers, we saw that if you stretch the muscle out, it's going to have more of a contractile sort of effect. If I were to shorten the muscle, it's going to have more of a relaxing sort of effect here. This is only going to be inhibitory with the tendon. The Golgi tendon reflex. And basically when it's going to detect changes in the muscle tension itself, so again, imagine you were to have the muscle start to stretch out because it's trying to contract here. So again, when you have the muscle actually contracting in the middle here, because again, it's all going to be going in towards the center, you're going to find that this is going to activate stretch reflexes here in the Golgi tendon apparatus, and this is then going to send a signal, depending how strong that stretch is, to eventually cause an inhibitory effect on the muscle itself, right? So again, a lot of times you can find that say things like the quadriceps are much stronger than what you may think it is, but you'll have these reflexes that will prevent you from basically ripping out the tendons, essentially, which is typically a good thing, right? Usually, for the most part. Some of these questions you should just say, yes, absolutely. They're not all rhetorical. Anyway. Okay, some other reflexes we can find. These are actually kind of interesting, I think. So here's this flexor reflex that occurs here. This is actually elicited when you have pain reflexes. This is one of the more common ones, common reasons why you would have this sort of reflex here, where basically when you have pain being sensed by one appendage, it's going to, and again, say, imagine like you put your hand on the stove. What's your natural reflex to do? Or at least a hot stove one that's actually on, I should say. If it's a cold stove, probably like, that's fine. But if it's a hot stove, it's on, what's that reflex going to be? Pull your hand away, right? So again, that should be the first reflex you're going to expect there, right? So again, you're going to have, say, the biceps are going to contract, they're going to deflex, and then your triceps are going to extend by relaxing, right? So again, it's important that these antagonistic muscles are working together, right? So again, if I had both the biceps and the triceps both activated at the same time, what happens to my arm? It's going to stay on the stove, and I'm just like, I need to get away from the stove. What is wrong with me? So in those cases there, you're going to see this reflex, or initially, you're going to have a reflex of pulling your arm away, okay? And what's interesting is that you actually have the opposite reflex in the other limb. So you can kind of see here, through the sensory input, through all these interneurons that happen here, some of them are going to cross over to the other side, and you can actually see that it will cause the opposite reaction in the other limb. Okay, so instead of causing my bicep to contract and causing my tricep to relax, it's actually going to cause the opposite. The bicep's going to relax, the tricep's going to extend, and that's going to do what to my arm, the other arm? Basically kind of pushing myself away. And again, I kind of think of this better example, I think, is like if you step on a nail. Say you're walking along, I step on a nail, say I step on it with my right foot, the right foot's going to want to do what? Lift up to get away from the nail, but then I have to support myself with the opposite leg, and so that's where that kind of antagonistic effect is going to be useful, because again, that keeps me upright instead of falling over just because I stepped on a nail, right? So again, it's kind of one pathway, one reflex pathway you can see there is kind of interesting. And again, different responses or different stimuli can actually elicit these responses. Here, pain is one of the most common ones. Okay, so any questions on those reflexes so far? Fantastic. All right, so let's talk about cortical and brainstem control of motor functions. Let's say, okay, we have those nice reflexes there, which are good for a lot of things, good for kind of quick changes. However, if we want to have actual control over what's going on in our body, if we want to have voluntary control of the muscles, what's happening? So, guys, remember the somatosensory cortex we talked about back all the way in section 1? We talked about the senses, like proprioception and nociception. What's nociception? Yeah, pain detection, right? So again, remember the somatosensory cortex is going to be located right here on the cortex. Now, it's important that it's right there because it's actually going to be interfacing pretty directly with the motor cortex as well. And if you're going to see, there's a couple different areas of the motor cortex. You're going to find there's going to be the primary motor cortex. Remember that weird homunculus looking sort of picture we looked at? It's going to come up again. You're going to find that the same areas that get represented by the somatosensory cortex are going to be represented in the same areas roughly, just a little bit further anterior with the primary motor cortex. We're also going to see there's going to be a premotor cortex and also a supplementary motor cortex. They have their own individual function as well, which we're going to talk about in more detail. And again, we're going to find there's a lot of input signals that are going to be coming up into the brain. They're going to get processed, especially through the somatosensory cortex here, and that information gets fed into the motor cortex. And then what happens from there, do you think? and then send signals back down to eventually cause muscles to do things, right? So we can say, okay, well, if I want to walk, that signals can come in and say, okay, here's the situation, here's our proprioception, we know we're standing already, let's go ahead and initiate the movements to walk. This is how that kind of process is going to go. Again, you're constantly getting inputs coming in telling you the state of things, and you're going to be sending out these outputs as well back to the muscles to actually initiate actual movements, okay? It's going to get more complicated than that, but that's kind of the general gist of it. Again, typically you're going to find that most of the signals that are coming from the CNS are going to interact with a lot of the interneurons. Very few of them interact with the motor neurons themselves. There are very few, but usually those are going to be for things for very fine control. So especially like the hands and the fingers, when you need to have really fine control of those muscle fibers, that's where you can actually have direct innervation. But generally, it's going to be connecting with the interneurons within the spinal cord. Okay. All right, so looking at the primary motor cortex, this is good for basically kind of gross movements, right? Not like gross movements like picking your nose or something, but like movements like of a gross nature of like, large movements of the body, right? This is not for like fine control of like saying, I want to change a light bulb or I want to suture or something like that. These are going to be kind of more gross movements. Again, a lot of the signals from, say, the supplementary and the premotor cortex are going to be kind of fed into the primary motor cortex, but this is good for basic kind of large movements here. And you can actually find you can excite, various patterns of separate muscles depending on how things are being activated here. But again, remember, what's kind of the thing to note about this crazy homunculus looking kind of guy? The larger areas typically represent what? More neurons associated with it, which means better control, right? So that way I know with the hands, I typically want pretty fine control of the hands. That way I can do things like suture, potentially, not me, but you guys will, or things like, the face, right? So that way I can, move my lips and tongue and all of that to make sure that I can speak to you guys effectively or ineffectively, some of you may put on my evaluation, who knows. But again, notice the larger the area here on the brain on this map, means the more neurons are associated with it, the better the control we have, the more fine control we're going to have of those particular muscles here. But things like, the feet, you probably don't need a lot of fine control of the feet unless you have like, monkey toes and you can pick stuff up off the ground. It's a lesser known skill of mine, but you may find that's typically less neurons associated with that than some of the other areas like the hands. Okay. Again, the premotor area is going to have a similar topographical organization. So just like we saw on the last page there, as the primary motor cortex, typically though you're going to see this is going to be more associated with more complex movement patterns, right? So movement patterns of associating or using different muscle groups and things like that. So for instance, if I need to like position my shoulders a certain way to get ready for a certain action, this is where the premotor cortex can kind of come up with this, right? And they kind of develop, we call this this motor image, where it's kind of starting to get the idea of, okay, what muscles do I need to start activating in what order in order to get this this event to occur, right? So I need to switch over to grab my mouse in order to go to the next slide. That is what's going to start to happen here in this premotor area, okay? Again, not as fine of controls, we're going to see some other areas, but still, this is where we can kind of get that motor image starting to be developed there, okay? All right, next we have the supplementary area, and so you're going to find that certain areas of the brain are located with different types of skills, as you'll see. So again, we're getting more and more specialized as we kind of move more anteriorly, and so we're going to see things like hand skills are going to be here in the supplemental area here. We can see things like head rotation, contralateral eye movements. Those are all going to be much more fine controls, much more specific movements we're looking for that get taken care of in the supplementary area here. Again, typically you're going to find this is also more associated with kind of bilateral movements. So again, if I'm having the eyes move to focus on something or having the head move, things like that. And typically they're going to be working with the premotor area for kind of body-wide sort of attitudinal movements, not like giving someone the finger, that kind of attitude, but like attitudinal is like kind of moving position, getting in different states in order to get a movement to occur, right? I like the attitudinal joke. Oh well. So how do these transmissions actually get from the cortex to the muscles themselves? We're going to look through both the corticospinal and also the, or otherwise known as the pyramidal tract. Okay, you know where the term pyramidal comes from? Yeah, pyramid, basically, right? So we're going to see the pyramids here in the medulla. It's as simple as that. Sometimes they just name things like that for ease of use, I guess. So you can see there's going to be these pyramids here in the medulla, and you can find they kind of have a pyramid sort of shape. Again, I'm not Frank Natter. I can't necessarily say that it looks exactly like a pyramid, but probably close enough, right, for people who are initially dissecting individuals. Anywho, but again, this is the main tract we're going to be finding here, and you're going to notice there's some accessory pathways as well. So we're going to talk about these individually, but things like the basal ganglia, things like the cerebellum are also going to be involved here. But this is kind of the most direct pathway that you can have for the most part from getting signals from the motor cortex down into the muscles to cause something to happen, some sort of movement to occur. And typically, the more direct the pathway is, the more detailed or more discrete that movement is going to be. So I mentioned some of these pathways are going to be interfacing directly with the motor neurons. Again, they're very few for the most part, but some of them are going to directly innervate that. That's good for fine control, like the hands and whatnot, right? And again, you're going to notice here as well that you're going to see this sort of kind of contralateral sort of crossing over effect that happens here. One thing to notice here is you're going to have this lateral corticospinal tract. This is where it is actually having crossing over occurring here. So again, the left side of the brain is going to control stuff on the right side of the body. But you're also going to have this ventral corticospinal tract. We'll talk about it a little bit. This is going to be staying on the same side. Okay, so just one thing to note there. Yes, sir.
So with the motor neurons, they cross over at. So it's not the medulla oblongata, or?
Yeah. So right here, so yeah, right. So once it's kind of passing through here, I guess this is another kind of a lower portion of the medulla there, right? But again, these are not the actual motor neurons themselves. These are just going to be these neurons that are carrying motor signals down to the motor neurons that are going to be actually in the spinal cord that will then interface with the muscles. Okay. Okay. So looking here, you're going to see it's going to be heading from the cortex, usually from, say, places like the primary motor cortex, going to be traveling down. And again, I'm not going to get specific, like it goes from here to blank to here, and you guys fill in the blank for a test. I'm not going to get that kind of granular with it, but kind of know the general flow of things. That's going to be traveling down from the cortex, eventually into the medulla there, right? So again, it's going to get those pyramidal fibers. This the only reason why I note the pyramidal fibers here, because we're going to talk about extrapyramidal tracts later on, and that's just going to mean anything that's kind of not traveling to this pyramidal tract. Okay, so we'll talk about that a little bit later, how that's going to be important, especially when you get to things like Parkinson's and other sort of really complex movement patterns. But anyway, again, once it's going to be traveling here, you're going to find that a lot of them are going to travel, they cross over and going to go down the lateral corticospinal tract, which you can see right here, and then they can terminate on to the interneurons. Right, and we just mentioned interneurons are going to be kind of the middlemen there, where they're getting a lot of different input signals to eventually interface with the motor neurons. There's going to be some sensory relay neurons that they can interface with as well. So again, the signals that are coming in, say from the muscles or from the skin or in different things like that. Or they can cause actual innervation of the motor neurons themselves. This is pretty rare, but it can occur, and this would actually cause direct contraction at that point. Okay. All right, so let's look at kind of the incoming sensory fibers to the motor cortex. What are all the different inputs that the motor cortex is going to get to make decisions on, well, what kind of movement should we start to initiate here, right? So you're going to find that we can get fibers coming from the somatosensory cortex, which I kind of already mentioned. We're going to have things like auditory and the visual cortices are going to be connecting here as well. Why is that important? Yeah, what I hear and see can also interface with what I'm feeling in order to make sure that, okay, I know what's going on, right? That's why I always see people get they lose their equilibrium. We'll look at the vestibular apparatus here in a few minutes. But again, when you put it like on a VR headset and you have someone you kind of tapping from behind, because their eyes and ears are not matching up with what they're actually feeling from things like proprioception and whatnot, they get very, very discombobulated, oftentimes fall over, and it's no good, right? That's why it's not fun to play with your friends who are in VR headsets at the time. But anyway, because again, that's also why you get a lot of nausea and vomiting that happens from that, because again, what you're feeling in the body is not matching up with what you're seeing and hearing, as the case may be, right? So that's why whenever you have kind of these inputs that are not matching up, that's when you end up getting things like nausea and vomiting that can happen from that. Anyway, so again, we're getting things from the somatosensory cortex, the auditory, visual cortices, and also the frontal cortex. Again, it's kind of our command center there, kind of we're making all executive decisions. You're going to have things coming from the corpus callosum, coming from the opposite hemisphere. So now we're going to be getting inputs from happening from the other side of the brain. So that way we can make sure the left hand's talking to the right hand, so to speak, right? We're going to have fibers coming from the thalamus. So this is going to help us with things like muscle signals, joint signals, some of the cutaneous tactile signals can all be kind of filtered through the thalamus there as well. If you go back to your first section of slides and look at those tracks, you'll also see the thalamus is important for that. And then we'll also get some information come from the basal ganglia cerebellum. This is also going to be really important for helping us to make sure we're sequencing movements correctly, making sure we are going to make sure that if I go to try to put my finger on this edge of the desk, I'm not going to go over it or before it. We'll talk about the cerebellum a little bit later on. And then also we're going to get some inputs from the thalamus. Is going to help us with like levels of excitability, so to speak. Okay. So again, this is just an idea of just all the different inputs that are coming in to make us aware of what's going on. So that way our motor cortex can kind of respond accordingly. Okay. All right. So some other tracks we're going to see here include things like the red nucleus. This is also going to be the start of what we call the rubrospinal tract. So rubro, and just prefix, I don't know what language it comes from, but it means red, so you can kind of think red nucleus, rubrospinal tract. And basically this is kind of an alternative pathway for sending signals from the cortex down the spinal cord, okay? Usually found in the mesencephalon, which you can see right here. This is not used, it's actually underdeveloped in humans compared to, say, other animals, but you're going to find this is very good for having discrete movements, very precise sort of movements here. You're going to find this is good for like fine finger control or hand control. So again, pretty underdeveloped here for humans, but it's still an important tract there to make sure we have good fine control. And honestly, one of the things you're going to see, and the reason why we know so much about what these different areas do is basically by disease, right? So for instance, if I have a stroke that occurs here, right, in the red nucleus, what do you think might happen? It cause ischemia and it cause that tissue to be damaged. What could occur? Yeah, lost fine control of the fingers. Maybe I had someone who's a classical violinist and now they can't perform anymore because now they lost that fine control. So again, very similar. Whenever you have any kind of infarcts or damage being done to the brain, there you're typically going to see those areas are going to take a hit, and it very clearly will be manifested in whatever their deficits are after the case. Anyway, so again, red nucleus, good for those fine kind of motor controls. All right. So going through, we're going to see that looking at the dynamic and static signals that get sent by those pyramidal neurons, right, and those are going to be in the medulla, going to be getting sent down. You're going to find, again, we have a higher proportion of those in the red nucleus as well, because, again, those are really good for those fine controls. So that way we can sense those changes, minute changes in muscle length and things like that for those dynamic and static signals. But basically, all this somatosensory feedback is going to come into the motor cortex, and this is going to help us control that kind of precision of the muscle contraction itself, right? So we're getting signals from places like the muscle spindles. We're getting signals from the tendon organs and other tactile receptors to give us a very fine idea in the motor cortex of exactly how the muscles are moving, what they're doing, so that way we can send signals back down. They're going to be very precise to make sure we're moving it as we want to, right? So again, that feedback can either enhance or inhibit the muscle contraction, right? Depending on what's going on. Like for instance, if I realize that, okay, the quadriceps is getting much shorter than it should be, you know, I need a stronger signal to make sure it lengthens back out as appropriate, right? Or whatever the case may be. So again, you can find, again, as muscle spindles stretch, again, that feedback, not only does it feed back into that spinal reflex, it can also feed back up into the cortex as well to send the signal back down. So you can see there's multiple tracks here, which is why all those interneurons are really important to make sure we have all the signals being sent the appropriate way, okay? Again, you can kind of see the signaling that happens here. Again, these are going to be the corticospinal tract from the pyramidal cells. This is going to be the main thing that's going to be coming down here. But again, here's that rubrospinal tract we talked about. And here's also this reticulospinal tract. Anything that's outside of the pyramidal tract is considered to be extrapyramidal. So whenever I say extrapyramidal, that's what I'm referring to. So kind of keep that in the back of your mind for right now, okay? So this yellow one would be the pyramidal tract. All these other ones would be extrapyramidal tracts. Notice here they're going to be feeding into these interneurons and then feeding out to the motor nerves themselves, okay? Anyway, all right, so moving on, there's also some other control of our muscles that can occur here with the brainstem specifically, and really the brainstem is kind of the control center for a lot of things, right? We kind of consider this the way station for command signals, especially with the signals coming up, kind of giving us a lot of information, and then we're able to kind of counteract and send signals back down. So things like control of respiration, right? So what kind of muscles do we control with the respiration? Or, yeah, the diaphragm's gonna be a good one. What else? Intercostals are going to be important there, right? So again, a lot of accessory muscles as well. So this can be regulated through things like the brainstem, cardiovascular function, GI function, etc. So a lot of different control here. Equilibrium is another big one. Control the eye movement. So again, all these can be helped and controlled through the brainstem here. You're going to see that there's several different nuclei where a lot of these signals are going to be coming from. And these are going to lead to their own tracts, as we'll see here in just a second. So we have, like, say, the pontine reticular nuclei, vestibular, and then the medullary reticular nuclei. We're going to look at their function here in just a second, but they're located within the brainstem and then signaling down, and we're going to see what some of those functions are going to be here. So first off, you can see here within the spinal column, you can see where we have, say, the pontine reticulospinal tract, we have the lateral vestibular spinal tract, and then also this medullary tract here. So notice it's going to be kind of more lateral here. And so basically the first two we're going to talk about is the pontine and then the medullary reticular nuclei. And so these are actually antagonistic to one another. So we're going to find that typically the pontine reticular nuclei is going to have a stimulatory effect on anti-gravity muscles, which, I mean, anti-gravity muscles are just what? Things that keep me standing up straight, right? So I can't make myself float yet. I'm working on it. But right now, the anti-gravity muscles are keeping me standing up against gravity itself. However, we can have things like the medullary reticular nuclei, which are actually going to be able to relax these muscles. So say, for instance, if I want to go from sitting to laying down, you're going to find that the pontine reticular nuclei says, hey, this isn't good. We need to stay standing up, because this is what its signal is saying. However, if the cortex can come through and say, hey, we want to override that, we want to go ahead and stimulate the medullary reticular nuclei, that can then overtake. It'll have an inhibitory effect and allow me to go from standing down to a laying down sort of position there. Okay, so that's one thing you can see. Now there's also a vestibular nuclei, and based on its name, what do you think it helps us with? Balance, equilibrium, and so it's going to be getting a lot of signals coming in from the vestibular apparatus, as we know, right? And so these are going to be working in association with the pontine particular nuclei. So again, and we saw that the pontine typically has a positive effect on these anti-gravity muscles. What's important is me working with the vestibular apparatus to make sure that we can help to maintain those signals there, right? So again, this is good where if I have, say, something where I'm like turning my head or I'm having some sort of motion occurring there, the vestibular apparatus is going to be able to pick up that, right? So I'm not going to belabor these points, but remember we talked about those kinecilium and stereocilium and all of that were being moved when you have different motions occurring within, say, like the semicircular canals and things like that. All the signals are being sent back and again going through and working again with the pontine reticular spinal tract in order to help us make sure that we can maintain that balance, make sure we can have our anti-gravity muscles working appropriately, so that way if I'm turning, I'm not just going to keep turning and fall over and that's it, right? Okay, up next we have the cerebellar and basal ganglia motor control. Get a little bit more complicated. Anyone didn't think it was complicated enough, but that's okay. So the cerebellum is kind of an interesting thing. We didn't know what it did for a long time. It's kind of what they call it the silent area of the brain for a while. However, what we do know about it now, it's very useful for helping us in basically sequencing movements, right? So if I need to sort of get a sequence going from one thing to the next, this is where the actual cerebellum can be very useful for this. I'll also have a smooth progression. Actually, you can see in a second here what happens if you don't have a functioning cerebellum, some of the problems you run into. So I'll talk about that in a moment. But very useful for making smooth progression from one movement to the next. Okay. And it can also help with doing things like making corrective adjustments. You're going to find that the cerebellum is good at learning certain motor patterns. So when we talk about things like muscle memory, this is where the cerebellum can kind of come into play to some degree. And I'll talk about that in just a moment here. And again, sequential movement. So again, if I'm going from a laying down position to standing up position, how am I going to sequence that? What muscles am I going to contract in what order to make sure that I have that smooth progression of sitting my legs out of the bed, sitting up, and then actually standing, right? And again, it's good because it learns from mistakes. You're able to make sure that if you can't do something right the first time, you learn from that mistake and then you can kind of correct it. We'll look at how that actually happens here in just a few moments. So looking at the actual topography of the cerebellum here, we're going to see you have the vermis, which is going to be located right here in the middle. This is where we're going to see a lot of functions for, say, like the axial body, shoulders, hips, neck, things like that are all going to be located right here in the center. You kind of see kind of similar to that homunculus sort of picture we saw with the motor cortex. I think this is going to be kind of associated here. You can kind of see the axial sort of body. The intermediate zone, which is going to be this blue area right here, is going to be more dealing with contractions of like the distal portions, the upper and the lower limbs, which you kind of see. Again, it's not as clearly depicted here, but you kind of get an idea. And then the lateral zone is going to be able to help with that sequential movement patterns, right? So again, going from point A to point B to C to D, make sure you don't accidentally skip from A to D and go back to B and do it incorrectly. Okay, so there's a lot of input pathways. Now, I'm not going to ask you specifically on the test, is this responsible for the cortico-ponto-cerebellar path? I'm probably not going to get that granular, right? I just want to kind of show you just how complicated some of these things can get by just showing you the different input pathways that can come in and how they can have different functions within the cerebellum itself, right? So, for instance, we're looking at something like the olivocerebellar tract. There's actually an inferior olive within the cerebellum. I only like superior olives, but they have an inferior olive here. And so you can see this is going to be excited by fibers coming in from the cerebral motor cortex, the basal ganglia, the reticular formation, the spinal cord. A lot of signals are going to be coming in. We have things like the vestibular cerebellar fibers, be coming in from different pathways. And again, just basically showing you that all of this information is going to be getting fed into the cerebellum, and then we're going to have an output. It's going to help us to do things like making sure that if I want to point at a specific point, I can make sure that I go and I touch the tip of that, and I don't go past it, and don't go before it. I can sequence movements appropriately, things like that. And looking at some of the afferent pathways from the periphery, you can start to see some of these signals are going to be traveling up through the spinal cord. Again, there's a dorsal and a ventral spinal cerebellar tract. This is going to be able to help us with things like, what's going on with the muscle spindles or the spindle fibers, what's going on with the Golgi tendon apparatus, different things like that kind of help us get an idea of, okay, what's going on as far as like position of the body, what are the muscles doing, etc. So we're getting a lot of these tracts are coming up. Not only are they going to the cortex, but they're also going to the cerebellum additionally, right? So we can send these signals in different pathways to make sure everything's kind of getting signaled appropriately. Okay, so looking at what's going to be sending out through the cerebellum, we're going to have these kind of 3 deep cerebellar nuclei. There's a dentate, an interpose, and a vestigial. And basically, they're going to be sending out different pathways here. So again, there's going to be one that can help with things like reciprocal movements of antagonistic muscles and peripheral limbs. Okay, so what does that mean? Basically, it means that if I'm going to try to, say, point out my finger and touch this, I need to make sure that I'm going to be loosening the bicep, but I can also contract the tricep to make sure that these antagonistic muscles are working appropriately, but maybe I don't go too far, right? Make sure that I can go ahead and have appropriate effects with the antagonistic muscles to make sure that whatever I'm intending to do, from say the frontal cortex, actually gets done appropriately, right? And a lot of signals can also go to the cerebellar cortex. There's a lot of kind of overlap of information coming in. So the cortex is also getting signaled by the cerebellum, not just directly from, say, the spinal cord specifically. Okay. So this is one of my favorite slides to try to explain because it looks like total gobbledygook, but it kind of makes a lot of sense when you kind of think about it. So if you think about, basically, this is going to be the output from the cerebellum. This is going to be basically the cortex of it itself. You're going to see there's kind of a nice sort of organized sort of pattern to it, where basically you have all these neurons that are going to be here. They're going to have inputs onto basically the nucleus here. This is kind of the nucleus of we saw the 3 different nuclei there. They're going to have the output signals. These are also going to be getting a lot of input signals that are going to be coming in from various places. So for instance, you're going to have these mossy fiber cells that are going to be sending in a lot of input from the spinal cords. The thing to note here is that one, you're going to have a direct input signal, a direct positive signal here is going to happen on the output nuclei. Okay, so again, all the inputs coming in are going to have a direct output on the nuclei, or a direct positive effect. You're also going to see this going to kind of travel up and kind of interface with the cortex here. And you have these Purkinje cells. Okay, it's different from the Purkinje fibers in the heart, but these are Purkinje cells. And you notice here they have a what sort of effect? They have an inhibitory effect. So again, any signal coming into the cerebellum is initially going to have a positive effect on the output, so that should initiate a movement. And then it's going to take a split second later, because this is going to take a little bit longer, the distance is further, it's going to travel up and eventually send a signal that's going to activate the Purkinje cells, and then eventually do what to the output? Going to shut it off, right? So again, it's just going to be excitatory, and then it'll have a split second later have an inhibitory effect. This is why we don't overshoot things when we're trying to say point. So again, this is a very specific point. I'm touching it. I don't overshoot and I knock it over, as the case may be. But I can touch a specific point without overshooting it or undershooting it, because initially I'm saying getting that excitatory signal is that, okay, go ahead and put your finger out, but then don't go too far, because a split second later you have those Purkinje cells that are inhibiting the output of the nuclei there. Okay. So it's kind of interesting. It prevents this kind of this oscillation that occurs, and we'll look at some of the problems that happen with patients who have cerebellar injury. So say they have like a cerebellar stroke or something, we'll look at some of the problems that happens from that. But a lot of it goes to, can they actually go and can they actually put their fingers to their nose, or can they do sequential movements like flipping their hand back and forth, right? These are some of the tests you're going to do to see if patients actually have good cerebellar function. Again, this is also where we're going to see where if we have say for instance, like you're learning to play an instrument for the first time, getting things down like the fingerings on an instrument, or like if you're trying to learn how to play a chord on a guitar or something like that, initially you can look at a page and it will tell you how to position your fingers, but you don't really have a good way to reproduce that initially, right? You have to learn that muscle memory, and so this is where a lot of kind of doing things incorrectly the first time, the cerebellum is going to be able to pick this up and say, okay, this was wrong the first time, let's go ahead and try to improve it the next time. So eventually, next time we try to make a C chord, it's going to go a little smoother, and go smoother and smoother till eventually you can just make it just for muscle memory itself. Okay, so that's another big part that the cerebellum is going to be playing here. So what can happen if you have, say, a dysfunctional cerebellum? You can find that you have what we call a dysmetria and ataxia. So basically by having, and again, this is kind of shown in either patients who have, say, like a lesion in the cerebellum, or if you have like an animal had that cerebellum removed, this can prevent prediction of like how far movements will go, right? So that's one of the things where if I'm trying to put my finger and I expect it to be right here, but actually go that far, right? Things like that. Prediction is not going to be able to happen appropriately. We call that a lot of pass pointing, right? Because maybe I don't activate the correct antagonistic muscle to stop my arm from outreaching. It's one example. Then more than it should. Other things like dysdiadokinesia is basically the inability to perform rapid alternating movements. So a good way to test for this in patients is to have them kind of put their hand front and back, right? So you guys can probably all do this, correct? If you have a patient, they would actually not be able to sequence that appropriately, and then eventually they wouldn't be able to really kind of get they might be able to do it for a few seconds, but then it would kind of die down and they would have no real good ability to do that. So that's one thing you can actually test for cerebellar function. Other things like dysarthria, maybe they're not able to talk appropriately, and then they can also have what we call intention tremor. That goes back to that oscillation of effect where, again, if I was trying to say go and touch the tip of this, but accidentally going to go back and forth, they end up developing this tremor there because they can't really inhibit that oscillation that happens there because they don't have those Purkinje fibers to try to inhibit the output from those nuclei. Make sense? So again, sequencing of movements, trying to make sure we can use the antagonistic muscles appropriately to make sure that when we go to touch something, we touch it and that's it, and they don't go past it, don't keep oscillating, etc. So it's main functions of the cerebellum. Okay, up next we have the basal ganglia. So if you thought the cerebellum was confusing, guess what? The basal ganglia gets even more confusing, as the case may be. But this is going to be made up of several different, trying to think of the word here, different areas of the brain here. It's going to include things like the caudate nucleus, putamen, the globus pallidus, the substantia nigra, and subthalamic nucleus. Okay, it's probably not as important you remember every single portion of that, but just know this is all going to be making up the basal ganglia. Okay. As I mentioned, the inputs and outputs that happen here are quite complicated, right? So again, some of these signals are going to be inhibitory. The dashed lines are inhibitory. The solid lines are excitatory. And again, this is all very, very complicated as far as the inputs and the outputs go. Okay, but what does the basal ganglia actually help us? Well, this is some of the most complex sort of movement patterns that we can actually do, right? So very complex motor activity, including things like writing letters of the alphabet, right? So being able to actually write letters appropriately, cutting paper with scissors, right? No, we don't. Some of us have to use safety scissors all the time because maybe our caudate putamen is not working appropriately. Who knows? Hammering nails, things like that, right? So again, very, very complex motor patterns are going to be handled through the basal ganglia here. Okay. You're going to notice that not many of these inputs are going to go directly to the primary motor cortex, but more often than not, they're going to be heading to the primary and the premotor. I believe I should put. I'm sorry. Not many inputs coming into the basal ganglia here, but a lot of the outputs coming from the basal ganglia are going to go onto the premotor cortex and also the primary cortex. So again, they're going to be initiating those movements, say, hey, let's draw the letter A, and then sending that out to communicate with the premotor and the motor cortex to go ahead and say, okay, well, this is the movement pattern I need to initiate here. Okay. So what happens if you have a dysfunction, you may have the inability to, say, write. It may have an inability to actually write appropriately, right? And so this is actually one of the things you can see with patients. I'll talk about Parkinson's disease in a second. This is the main disease that affects the basal ganglia, but you're going to find that they have things like dysgraphia, where they can't actually write letters appropriately anymore. The size of the letters isn't appropriate for what they're actually trying to write. It can affect the arm, the neck, the face. And they also see this kind of flicking movements that can occur, these chorias, because the patterns here are not going to be done appropriately. This looks like in a second. Other things you can see with the caudate circuit, part of this is going to be important with cognitive control of motor activity. And so you can see issues here where, this is more kind of complex patterns. Things are going to take more than, say, like 5 seconds or so. For instance, I imagine if you're having to do something like respond to an outside threat or something. So in the case of, like, say, a bear attack, how do I sequence the events that I need to do to actually get away from that bear? So, okay, if one bear pops out at me, I need to know to turn around. I need to know to start initiating the running pattern. I need to make sure I start activating that pudendal nerve so I don't pee my pants. I need to make sure that I know how to reach out and open the door so I can run out and close it behind me so you guys are all stuck with the bear. And then, so again, a lot of these complex movement patterns, and usually more than 5 seconds or so, are going to be handled through this caudate circuit here, right? Or for instance, imagine if you have, say, 100 people or so that are egging you on to go play ping pong, even though you have no right in the world to be playing ping pong. I had to tell my brain to say, okay, put on your big boy face, stand up, go out there and actually try to play ping. So again, a lot of complex movement patterns handling through the caudate circuit here. Okay, so what happens when you have damage done to the Putamen circuit and the basal ganglia is you can actually find loss of these dopaminergic neurons. Basically, these are neurons that are releasing dopamine. We'll talk about this in the next section coming up. But basically, these are going to be, you develop Parkinson's, right, when you have a loss of these dopaminergic neurons. And so you can see things like it's very difficult for them to actually initiate movements, right? Things like causing rigidity. They can also have problems where they have difficulty kind of initiating movements, and so they have to kind of like shuffling gait. It occurs, I don't know if anyone's ever dealt with someone with Parkinson's before, but this is kind of very common things you run into. They also have things like involuntary tremor. They have like pill rolling sort of motions that they'll make, all related to the fact they don't have these dopaminergic neurons that are able to initiate movements appropriately, right? Has anyone ever seen the movie Awakenings with Robert De Niro and I don't know if it's Robin Williams, it's someone else, I think. No, it was a movie. It was about Parkinson's based. It's called Awakenings. Basically, it was about the discovery of using a certain drug that we use called Cinemet now, but basically of waking these patients up. If you ever get a chance, look at that movie or look up some clips from YouTube or something, but you get an idea, because eventually when you have enough damage done, they have no dopamine left, they become catatonic. They can't initiate any movements whatsoever. So the basal ganglia is very, very important for initiating a lot of different movement patterns, usually more kind of complex movement patterns that require several seconds of sequencing in order to get them done appropriately. And just to show you kind of the complexities that happen here, here's an example of what you would see with, say, like a normal patient. Again, we have things like the striatum, the globus pallidus, the substantia nigra, and again, all of these things are going to be complexing with the thalamus and then heading up to the motor cortex. And again, this is how things work normally. We're going to have things like GABA. We're going to talk about GABA in the next section coming up, and glutamate and things like that to make sure there's a good balance here. And dopamine is a really big component of this. Well, if I lose these dopaminergic neurons in substantia nigra, all of a sudden you're going to find you have a much more inhibitory sort of effect here, actually preventing movement from occurring in the motor cortex. Am I going to ask you anything about this slide in particular? No, but I do want you to know that this is some of the imbalances that can occur here, and why you would see with Parkinson's patients how they have a hard time initiating movements, whereas with other patients, say with a cerebellar hemorrhage or something, they have a hard time stopping movements, right, based on the different functions of the brain here. Make sense? Hopefully clear as mud. Teachers used to say. Okay, so that's it for skeletal muscle control. Any questions on that before I move on? Fantastic. So let's move on, and next we're going to talk about smooth muscle briefly. So skeletal's got the majority of the show dedicated to. Let's talk about smooth muscle briefly here. So smooth muscle, where do we find smooth muscle at? GI tract, around organs. Anything else? How about your hair, right? The follicles have smooth muscle associated with it for piloerection, things like that. So there's kind of two varieties of smooth muscle we're going to find here. We're going to have multi-unit smooth muscle. You can kind of see an example here. It's usually a lot less organized than you're going to find with the unitary smooth muscle, but these are going to be discrete, separate fibers that are all kind of working independently of one another. And they can be stimulated by neuronal control. This is mainly the case, right? So for instance, if you have a case where you need to cause piloerection, it's usually going to be under neuronal control versus we're going to see with the unitary control, this is going to be much more under hormonal control, as the case may be. So we'll see that in a few seconds here. But other muscles that kind of fall into the multi-unit smooth muscle include things like the ciliary muscle, the iris muscles, piloerectors, etc. Right? The unitary muscle is very syncytial. Anyone remember what that term means? All works together, right? There's a lot of gap junctions and things between these cells to make sure they're all working in concert. You don't want just one part of your stomach or say uterus to contract versus say you want all of it to contract if you're trying to get rid of a baby, right? Not get rid of, but birth the baby. I'll say that. So much positive spin on that term. Typically, this is going to be much more kind of uniform in shape and structure, such that you have kind of shapes or sheets or bundles of the smooth muscle here that provides for a good sort of, kind of concerted effort in order to get squeeze or relaxation, as the case may be. So you're thinking like GI tract, thinking the uterus, blood vessels, etc. Okay, this is going to be more of your unitary smooth muscle. So you're going to see that this is going to be in direct contrast to how the skeletal muscle is actually working. With skeletal muscle, it was striated, it was very, very regimented as far as how it was organized. Here's going to be a little bit more kind of amorphous, as the case may be. Here's some examples, some smooth muscle cells. You're going to still see that they contain actin and myosin filaments. So they still contain that, so that hasn't really changed from the skeletal muscle. But there's no troponin complex, right? So again, there's going to be no troponin complex that normally, what binds to troponin to activate it to cause ADP sites to be shown? Okay. Calcium, yeah, calcium had to bind to that, right? So calcium is still going to play a vital role to smooth muscle contraction here. However, you're going to find that the troponin is not going to be a constituent here, right? You're still going to see it relying on calcium, still relies on ATP, but the function is going to be a little bit different. So we're going to look at those in just a second here. So again, no striations. No really kind of organized differences here. However, what you're going to find is you have these dense bodies that actually function similar to what the Z disc did. And the Z disc did what in the skeletal muscle? Attached to the actin, right? So remember because remember the sarcomere is kind of divvied up into from Z disc to Z disc, and those were actually had the actin being attached to, and that was part of the I band, which is the light band that you saw there. Here you have the dense body. You see the actin filaments kind of attached to either end here, and you're going to notice the myosin filaments going to be right there in the middle. Okay. So also you're going to find the myosin filaments tend to have these kind of side polar cross bridges, meaning that it's not just sort of a flat sort of image here. You can imagine it's going to be kind of going all over, and so myosin can be pulling actin from kind of in all directions essentially to cause the cell to contract much more so than you would see say in a skeletal muscle cell. So for instance, here you can find they may contract up to 80% of their length. Skeletal muscle maybe only get, let's say, 30% of the length there. So again, these cells are going to be much more able to kind of contract in from all sides and get much, much smaller than a skeletal muscle cell would. Okay. So the goal here when you have smooth muscle contractions is to have a nice, smooth, prolonged sort of tonic contraction, right? You can have smooth muscle that's contracted for hours, days, weeks, years. You're going to find this is much slower cycling than what you're going to find with a skeletal muscle. Normally skeletal muscle, calcium gets in, causes a contraction, calcium gets out, and you're done, right? Can cause prolonged contraction, but we saw that due to the energetics of it, due to the fact that you need things like phosphocreatine and ATP and all of this in order to keep the contraction going, it's much more energy intensive. This is a much more efficient process when it comes to smooth muscle. It requires just a fraction of the ATP needed to actually keep a good strong contraction going for a long period of time, as we'll see here. Okay, about 1/300th of the amount of energy needed to maintain contraction as opposed to skeletal muscle. Okay. So looking at this, instead of troponin, we have a molecule called calmodulin. And so imagine here we have the extracellular fluid. Here's a smooth muscle cell. And we're going to have calcium. It's going to be coming in through the extracellular fluid. We're also going to have calcium coming from the sarcoplasmic reticulum. So in this case, this hasn't really changed from the skeletal muscle whatsoever. So we're going to have calmodulin. That's going to be interfacing with calcium. So we're going to see this come together. And you're going to activate what they call the myosin light chain kinase. So you see that MCLK, that's what that means. So basically, when I say a light chain kinase, what do you think that does? So kinase phosphorylates something. So again, good. We saw phosphorylation. We're still going to require energy here, as we'll see. But basically, what that active myosin light chain kinase is going to do, it's going to take ATP. It's going to break it down into ADP plus phosphate. But that phosphate is actually going to go and activate these myosin heads. It's going from inactive to the active state here. Once they get phosphorylated, at this point, now they can interact with the actin. Okay, because remember when we were looking at the skeletal muscle, normally the myosin heads themselves, they were able to cleave the ATP. They had that ATPase activity. This is much less so with the smooth muscle cells that you're going to see. Really, it requires that MCLK to come in and phosphorylate those heads. Once that is done, then they can interact with the actin and cause a contraction to occur here. Okay? Again, still really dependent on the extracellular calcium coming in. You're going to find that the sarcoplasmic reticulum is not nearly as developed as a skeletal muscle, but it's still an important thing here. And again, when we're looking at things like, if we wanted to say, say for instance, we're talking about blood vessels, right? And typically when you have smooth muscle contraction of the blood vessels, what happens to blood pressure? So squeezing the vessels, pressure should increase, right? So you have patients who maybe have hypertension. Well, what if I wanted to relax those muscles? What's one of those ways you think I could attack that? Maybe prevent calcium from coming in, right? So maybe I have a calcium channel, I have something blocking it. What would you call that? Perhaps a calcium channel blocker. And so something like Norvasc or amlodipine can actually do that. You can actually block this calcium channel here, and by preventing calcium from coming in, you prevent those smooth muscles from actually contracting, and guess what? Now your blood pressure goes down, right? So this is going to be a big set of drugs we're going to talk about when we talk about hypertension. We also see that calcium is really important for myocardial muscle as well, especially those pacemaker cells. So I also see calcium channel blockers playing a big role there too. So kind of things to start thinking about, keeping in mind. So how can I modify the system? What can I do to it to kind of bend it to my will to make sure I can deal with my patient's disease states, whether it be their sugar's too high, their pressure's too high, pressure's too low, whatever the case may be. So how do we kind of stop the contraction once we're done? So again, this is a very slow sort of process compared to skeletal muscle. Basically, you're going to have calcium pumps that still require ATP, pumping calcium back in to the sarcoplasm. Pretty slow though compared to the skeletal muscle, so it's still going to be slower process. Not as quick as the skeletal muscle. And then we're also going to have another enzyme here called myosin phosphatase. What do you think a phosphatase does? Just gets rid of phosphate, right? So again, we have a kinase that puts a phosphate on. We're going to have a phosphorylase, or phosphatase is going to take off a phosphate group. And so that's actually going to take the phosphate off of the myosin heads. So you can see these go back to an inactive state. And then eventually that calcium, as the levels are going down, as you're shuttling calcium back into the sarcoplasmic reticulum or out of the cell, and then you're going to find the calmodulin is going to dissociate and it's going to go back to kind of the resting state. So this is when you have a relaxation of that smooth muscle there. Okay. Notice here it doesn't really require as much energy as the skeletal muscle, so this is why it's much more efficient than you would see with skeletal muscle. Right? Now, how can we control this? So again, with skeletal muscle, how do we cause activation of skeletal muscle? Acetylcholine working on nicotinic receptors, remember? So again, that was all neuronal based, right? You had to have innervation of that muscle to release acetylcholine onto nicotinic receptors in order to cause those channels to open up. That allowed what to flow in? Sodium That allowed the cell to get to that threshold where it caused an action potential to occur, and then what opened up? Voltage-gated sodium channels. Good. So again, that is the way the skeletal muscle activates. We're going to find there's a lot of other ways we can actually activate or inhibit smooth muscle, as the case may be. So in this case here, say for instance with nervous control, we have things like autonomic control of smooth muscles. So we talked about this quite a bit. And think about the detrusor muscle. And when do we want to, say we have parasympathetic innervation of the detrusor muscle, we'd have what neurotransmitter? Acetylcholine interacting on what receptor? Muscarinic in this case, right? So again, different receptor types, but we're also dealing with different sort of muscle here, right? So again, we're dealing with smooth muscle. We're going to have muscarinic receptors being activated by acetylcholine that then causes constriction, right? So then that would be doing things like opening up calcium channels, allow calcium to flow in to cause that myosin light chain kinase to be activated via calmodulin. It's going to cause the myosin heads to be phosphorylated and cause a binding of it with actin, right? So again, these are things you can see there. This is under neuronal control. Now again, you can also have action potentials being kicked off by this. However, this is much less of an important role here. But calcium is going to be much more important for causing that spread of depolarization than the sodium would. But it's a lot slower because again, looking at a normal sodium level in the blood's what? Like 140 milliequivalents per liter, right? What's a normal calcium level? Do we know? It's like 8.5 to 10, somewhere like that. So there's a lot less calcium out there floating around. However, when you have calcium flowing in, it's going to be much slower than sodium because again, the concentration gradients are just that much bigger for sodium, right? So, remember, the bigger the concentration gradient, what did that do to flux? You saw more flux, right? Because again, the bigger the difference there, the more it's going to flow in. That's not as big of a difference for calcium, so you don't see as big of a change there. Okay. And again, looking at, say, for instance, just a single action potential coming along, you see it's going to be very quick. However, with smooth muscle, you're going to see much more long, contracted sort of, or longer, stronger sort of contractions with smooth muscle. And so you can see things like these slow waves that occur here. You might see this in something like the GI muscle, or you can see these action potentials causing, really prolonged contraction over a long period of time before it eventually relaxes. Okay. So again, this is kind of in direct opposition to the skeletal muscle, which is much a quick kind of one and done sort of thing. As long as it's getting action potential, skeletal muscle can stay contracted, but again, it requires a lot of energy, so you're going to find it's not going to be nearly as efficient as a smooth muscle. Okay. Again, you can find about 50% of that smooth muscle can be contracted or can be affected without actually having any action potentials interacting with it, right? We've already seen numerous examples of that. So for instance, looking at blood vessels respond to things like lack of oxygen or say excess CO2 or things like decreased pH, right? Say for instance, you have changes in pressure levels. Baroreceptors can affect blood vessels as well. And different things like that can all affect can affect how well those vessels are going to be either dilated or contracted, as the case may be. Hormones as well, things like epinephrine, angiotensin II, vasopressin, oxytocin. We looked at numerous ones of these that all affect the smooth muscle without actually causing any changes in action potential. A lot of them will just cause things like calcium channels to open up or cause other downstream effects to occur that will allow calcium to flow in and eventually cause contraction there. Okay. Again, some of these are going to affect the resting membrane potential. What do you think it's going to do? So say I raise the resting membrane potential, what does that do to the cell? Okay. It's easier to have an action potential, right? Because again, the difference between that threshold there is going to be a lot smaller. It's easier to get an action potential. So in some cases, it can either directly open ion channels to allow activation, or it can just change that resting membrane potential, right? All right, so any questions on that? Guys feeling all right? Can I keep going? Maybe? Okay, let's keep going for a little bit.